Apple Arcade. Yeah. 3 months. July.
Sorry, just get used to it. I'm not getting used to it. Stable. Everyone settled? Alright. Everyone okay? Shall we start? Make sure you can hear me. Yeah. Alright. Sounds better. Alright. Today is a new chapter. We talk about the bacterial growth. So there will be 2 lecture covered for the bacterial growth. And let us look at the content. And the first content that we want to know is the reproductive strategy. Very short, just a very short introductions regarding the reproductions. And different organisms may have using different strategy. And then the bacterials, they're using the binary fissions as the way for cell divisions. And then we go into this, which is the main content of today, and which is any environmental factor that affecting the growth. Right? So it's all come from the external factor, and we will talk about the one that highlight in blue. And at the same time, when we talk about each of the factor, how they limited the growth of the bacterial, or maybe also archaeobacterial. And at the same time, we also talk about how the bacterial or archaeobacterial, they adapt to this kind of the external factor. Alright? I shouldn't pass too near the mic, too near the front. But I need to, if I have a remote, it's better. Yeah. The one? I purposely find the one that cannot. Because I use the one that I always push the wrong button, so they always like, the slide is going. So I purposely find only one function for the laser. I should have a separate one from this side, but no. Which one should I look at? So for the learning outcome, right? As I said, that you may need to describe the bundle fission process, which important is a process, how the bacteria conduct the cell of the binary fissions, and then towards this one, you need to know what are the environmental factors that have the impact which affecting the growth of the bacteria, and also what kind of strategy that being used by the bacterial to actually survive in the presence of this extreme environment. Now, to start with, bacterial chromosome. Why I start with bacterial chromosome? That's because during the cell divisions, the DNA need to duplicate it, right? So the bacterial chromosome, we are not going into the genetic of bacterial genetics. That's why we only have one slide regarding to this. And all you know that they have the chromosome, but their chromosome is located to a region called nucleoid. They do not have nuclei, membrane-bound nuclei, but they do have a region called nucleoid that normally where they place their chromosomal DNA. And in order, because the DNA is really big considered in megabases, that they need to fold it, although but this is the circular shape of the chromosomal DNA packed into very small size in order to fit into this nucleoid. So it's almost like 1,000-fold, okay, folding it into small size. And looking at the, this is, you can calculate it, how many number of loop, this is the loop, they need to fold when they're forming the loop. And they have something, the protein, that at the center can actually adhere to each of the loop, that so it can look into this form. So then they further even supercoin each of the loops and then make it even smaller. So the entire purpose is to pack into a small size. And you can calculate that E. coli has around 50 to 100 loops, and each loop has 40 to 80,000 base pairs. And therefore, the genome size is about that, 5 megabase. So megabase is in 5 million base pairs. So when they talk about human has about 3 gigabase. So 3 gigabase is how many zero at the back? 9 0 at the back. This only have 6 0. So you can see humans has even bigger size of the genome or chromosome. So basically, consider 5 megabase is very small, very small. But bacterial cell is also very small. Although the size is small, but in order to pack into the small size of the cells, they still need to pack in this way. So this is a comparison. One that E. coli, this is E. coli chromosome. They are more or less look like that. This is the center, look like the center, and then the all other are supercoiled loops. And this is a eukaryote, which is normally we call chromosome as well, right? So the difference between eukaryote and prokaryote chromosome, one big difference, that this is circular. However, the eukaryote chromosome is non-circular. You can answer in this way. But to be precisely, they do not have, they have the end. They are linear. They have 2 ends. If you actually lose all of these chromosomes, they actually has one to the other end. They are linear. They are not circular. So they will be different in terms of during the replication because this is circular. The eukaryote chromosomes are not circular. All right. Furthermore, that is how the organisms they do the asexual reproduction. Here is the asexual reproductions. And when you talk about asexual reproduction, it's different from reproductions, right? Reproductions is the mating. Normally, you have in higher organism, you have different sexes, female, male. But in this case, asexual reproductions, basically, they are reproduced from, let's say, the parental body. So that's one big difference that genetically, their DNA are identical to the parental DNA. So like a clone, they are identical if they're doing the asexual reproductions. And you will see 2 very common reproductions mechanism. One is binary fissions, and this is binary fissions. You see this is dinophysis. It's one kind of dinoflagellate, and they are individual when they actually is mature. This is actually during the divisions, the cell binary fissions. So they will generate a new one, sort of like, they will generate in a way a new one. Yeah, so it looks like this. And see, the budding look like this. This is another called hydra. It's the animal. They bud at the region here. So there is a new bud coming from this region. So when the new bud become bigger and bigger, they look very much identical to this body. However, they are much smaller. So what happened after they have finished budding? They would detach from the body, the main body, and they become very small, and they actually attach to the ground. And then when is as big as this. Right? So this is budding. And again, genetically, they are identical to the parental body. So there are two. What other microorganism also doing budding? Yeast. Yeast is still doing budding. Bacteria is doing the binary fission, but yeast are doing budding. Alright. So, as you know that they actually, the bacteria, archaebacteria doing binary fissions, and one cell division, it takes 20 minutes. So, under the laboratory conditions. So it's very fast. So if you have 1 hour, it's almost like 2 generations from the bacteria. So that is because they are actually have a very short generations period of time. They are ideally, somebody has actually given a very good idea to study the mutation rate. So we know that all organism DNA can be mutated anyhow. But if you want to measure the mutation rate of human, no way. Because one generation, it takes like 20 years at least, right? I mean, minimum could be 15, 12 years, 13 years, but it takes so long to produce one generation. But E. coli, ideally, only 20 minutes for one generation. So the idea is that they have the original stock of the E. coli. So as a very origin of the stock, then they start to culture the E. coli. They keep the stock and then they take some of it to culture in the lab. Okay, after like 1 week or maybe 1 month later, they take a portion. They actually culture it every day non-stop. They keep it and a stock after 1 month and keep it in a stock, frozen it. Another month, you keep another stock. So all the stock I've been keeping up is 1 year has 12 months, they at least has 12 stock. So they do it continuously for more than 10 years. So after they have actually making millions of generations of E. coli, then they actually checking the sequence there, E. coli DNA. It's also not very expensive because E. coli, they have very small genome, easily been sequenced. And then they will see from year 1, year 2, year 3, how is the mutation rate going. So this is a very long project, but ideally it's a very good idea. And this diagram, I think this is the electron microgram. It's just been colored, and you can see this DNA actually also separating, and the 2 daughter cells also separating from each other. Okay, now, as for the binary fissions, this is a process. Anything they start with, they will start with, as you remember what I said, is the ORI, O-R-I. ORI basically is just a DNA sequence. But this DNA sequence, very specific, as has been recognized as a start site of replication. So when you say that ORI, and meaning that it's a start site for the replications in bacteria. And when they're starting with, they will start to replicate the circular chromosome. So how do they replicate? This is the origin, this is the site where you start the replications, indicate you should start here. So they actually, the DNA polymerase go 2 directions because you need to actually go in 2 directions, not 1 direction. Not 1 direction to go 1 round and do the replication. Actually, they do 2 directions, so they only go around for the half of them and it's done. Alright? Rather than you go 1 round taking longer time. So when they actually go 2 directions do you remember when you talk about DNA replication, you have a lagging strand and the other one? I forgot. Sorry? Leading, yes. So you have lagging strand, leading strand. That means that is 2 directions going. Alright? Now, during this time, when they start to replicate, you have this called this zig. This zig has a tubulin in eukaryote, this physics start to express a sample. And during this stage, right, now they're almost like done with the replications. So they're separating these two chromosomes. And the fixed zig protein, what they do, and during this, they actually start to assemble at the middle of the cells. And the cells now is already starting to elongate. So the physics is concentrated and aligned at the center. And here, where it's being aligned, then they have this cleavage vulnel that is created through the invigilations of the membrane. And here is formed a so-called fixed ring, which is very important. Without this, the cells cannot divide. So the fixed ring contains the fixed protein, which is the most important protein, plus many, many other proteins. And altogether, they also form the so-called divisome, right? And when you actually start to actually make it into two daughter cells, and of course, the fixed zig, when it is a ring, it becomes constricted, becomes smaller and smaller. So when it becomes smaller and smaller, and it separated them, right? And
Here.
Still can hear me? No problem, huh? So, after that, there is a sector regions that created. This septum, it called septum, and basically is the membrane. It's the membrane and the cell wall, the new membrane cell wall synthesized. And then they separated into two daughter cells eventually. You still can hear my voice, huh? Okay, should be okay. So do you clear about this? This is how they separate. Actually, it's already simplified a lot. Yeah, the actual fact is a lot of protein involved as well. Now, similarly, archaebacteria also doing the binary fissions. What happened is that the archaebacteria, actually this diagram taken from this paper. If you want to know a little bit more, you can check this particular paper. And they are using another protein system they call CDV. CDV has a homolog that found in eukaryote, which is called ESCRT, endosomal solid complex required for transport. A very long name, but basically ESCRT is the system that used to ubiquinated the protein, any protein that intact by ubiquin, and then it will be degraded into the lysosome. So those are the systems used for the ESCRT. However, this homolog had nothing to do, this CDV have nothing to do with this kind of mechanism. It has been used totally for different purposes. It's been used for cell divisions. So we're starting from here, 1. And it starts with this CDVA, recruit CDVB, they're working together, and then they form a polymeric ring at the cell center. It's very much same as the big zig in bacterial. They're actually forming a ring at the center. As you can see that the purple ring here, which has been stained with purple color, and indicate they are CDVA and CDVB. So the blue color is the DNA. Normally we stain the DNA in blue color because of the dye. It's blue color. The dye is not blue color, but it can be detected with UV light. That's why it's turned out blue. Now, this particular ring, then they recruit furthermore protein, CDBB1, B2. So these two proteins come along, and then B1, B2, and then they're going to remove this CDB A and B. How to do that? Then they come along with another CDBC. CDBC will be extracted, the CDBB, from the big complex, right? And so it extracting CDBB out of that and make it soluble, so that once it's soluble, it can be degraded by proteasome. So proteasome will come along to degrade CDBB, and then CDBB gone. So you actually remain the green color. The green color is the CD, B1 and B2. Now, similar like zinc, this CDB B1, B2 can contract. And then when they disassemble, they will losing part of it, the unit, and become shorter and shorter, right? When they actually disassemble, the entire contractor ring becomes shorter and shorter. So it's also contracting the cells. So exactly same as physics, and then it's smaller and smaller. Then all this CDB B1, B2 diffuse along the cytoplasm, and eventually you can. They also moving. Once they form two daughter cells, they also degrade the CDBC again. They removing their B1 and B2 out of it, and then you get two daughter cells. So this is how they do. Very similar binary fissions, and using those cytoskeleton protein to actually separate the two daughter cells. Similar in archaebacteria and bacteria, but however, remember, whatever the things that the protein they use, for bacteria it's physics, for this archaebacterials is using the CDB system, which involves several of the CDB protein. Alright. What is this? Oh, there is other. There's other bacterial which is not undergoing binary fission. They have their special ways, and it's worth mentioning it. And one of them is the Hypomonas neptunium, which is this one. This is on the, I think that picture is taken from the agar plate. So they look like this. This Hypomonas neptunium is very interesting because they have the advantage. This is the advantage they call, or stalk, right? They actually has the growth of the advantage coming out. And this, where they have this particular structure. Okay, this is for budding cells, but their particular structure is called biphasic prostate. So it's have this very special structure, and this name. No, what happened is that they are having the swarming cells. This is the swarming cells, meaning that they are free swimming in the ocean, and they have the flagella. So when they start to undergoing the asexual reproduction, the flagella will detach from the cell body, and then the cell body will have the growth of the appendage, or we call the stalk. So when they're growing longer and longer, at the other end of the stalk, there are something, that's the cell budding out from the other end with the flagella, right, growing. And once this has done, they will be separated. And for one, this is still becoming the swarmer cells, and they will be swimming around until they actually reach a stage. They will do another round of the asexual reproduction. For this, the one that have the advantage, have the stalk, they will actually attach to the rock or the ground and be like that. So that actually has two types of morphology. And for cyanobacterials, also interesting. They are very simple. They have their cells. So the cells are undergoing the DNA replications. So you have the divided into 2, 4, and then they accumulate a lot of this, right? They actually DNA replicated inside this extracellular matrix. So inside one extracellular matrix, supposed to be the cell membrane, but then they turn into extracellular matrix, and inside there are multiple round of replication. So generate a lot of these small ones. Eventually, the cytoplasm also being divided, and they call biocyte. Okay? So biocyte, once the biocyte has been accumulate, then they will actually break out and release. And each of the biocyte will eventually grow and become a vegetative cells, and will start the cycle again. So interesting is that they actually has to produce inside one extracellular matrix, one membrane, produce many of these biocyte, small cells. And streptomycin is the filamentous bacterials. And how do they grow? They have the fragments. So in one fragment, sometimes we consider as a cell membrane, but in one of the cells, they have many nucleoid. So they have multi-nucleoid in a fragment because the septum is a septofilament and septoridin, so they have many others. Now, why they have to do it this way? Because when they are branching out, growing, right, they have to make a very long filament, right, these vegetative cells. Now, when they want to make the spore, what happens that each of them which they will separating the nucleoid from each other by having a septum that produce. So then they make like that. Each of the individual of the cells from a cell, because they have been separated, will become the spore eventually. Okay, so there are many ways they actually carry out for asexual reproduction. Alright, we come to the main content here. What are the environmental factors which I want to include in this lecture? That about the solute and the water. The second one is about the pH, and the third one is the temperature, oxygens, and the light. That all these, right, they are the environmental factor that bacterials, in order to adapt this kind of environmental factor, they actually make adjustment to adapt to it. Very simple. This one is the water availability. What is water availability in the environment? We're talking about environment. The amount of water that present in the environment, right? But how do we know, how do we measure if we want to know whether this amount of water is enough for the growth of the bacteria, but they are in terms of percentage, let's say. And before we go that, we will osmosis. Osmosis is very simple. What happens is that, and if you have something that the solute can be sugar or any salt, right? A molecule. Normally, if they cannot passing this membrane, it's only water can freely go in and out. So if you have one side have a lot of salt, so the water flow will go through the directions. So we'll go to the one that have higher concentrations of the salt. Correct? So this is osmosis, the flow of the water. Before that, I think I need to actually it's not this one. It's not this one, another slide. So hypotonic, what happened to hypotonic? Hypotonic is a situation is that is a high concentrations of the solutes, which is can be the iron, sodium, potassium, whatsoever, that high in the cells than outside. So then the water will go into the cells. This is a situation we call hypotonic. Now, hypotonic will make the cell burst because if too much of water going into the cell bursts. However, good thing is that bacteria have a cell wall. The cell wall actually can maintain the cell shape, not allow it to further allow the water to go in. So it maintains its cell shape because of it's very rigid. There is another system called mechanosensitive channel, which is being used to allow the solute to leave the cells. When they actually too much of water going in, they will also pumping out the solute so that reduce the concentration of solute inside the cells and stop the water going in. So that's the mechanism to maintain cell size and cell shape. Hypertonic. Hypertonic, when the concentration, the solute concentration high outside the cells, but not in the inside the cells, meaning that water will coming out from the cells, and the cell will shrink, and then it's collapse. So this is the time we call hypertonic situations, and the cause of the cell strength and collapse we call plasmolysis. Eventually the cells lyse because they collapse. The membrane detached from the cell wall. You can see. Okay. See on this next one. Now, the first thing we talk about the water availability, and then we come to talk about what is aerophile. Water availability in the environments, how do you measure? How do you think that whether that is a water availability? I have two glasses here. Each glass I have, let's say, 100 ml of water. 100 ml of water. So the volumes of the water is the same in two glasses, right? Now, for one glass, I start to add the salt until it is fully saturated. So very high salt. Now, I'm asking you because there are different ones with salt, one without salt. Is there any change in the water volumes? Is the volume of water change? Yes? No? No, right? The volume of the water still remains the same because I didn't actually pour out the water. So the water volume remains the same. But what about the water availability? So the water availability definitely is different. So the water availability with the one just with the water without any salt is 1%. I think it's 1%, meaning that it's fully water. However, the one with the salt is all the water actually no longer able to dissolve the salt. So the water availability will definitely drop, but maybe drop to 0.2%, something like that. Now, that is different in the water availability. Same for the bacterials. Bacterials actually require water to survive. So if the environment have a different percentage in water availability, of course some bacteria require the 100% that's full of water, meaning 1% on water availability, yes, then they can survive. If the water availability drop, they cannot survive. And that's the meaning of water availability. However, most bacteria require water. Some bacteria require only a very little water. Those are the aerophile. Aerophiles, aero is salt, phile is lover, they are salt lover. Now, how do they do? Because in this case, aerophile, they are living in the hypotonic environment, meaning that there is a lot of salt, right? High salt at outside of the cells. So the water will tend to come out. However, they can able to maintain the cell shape as it is. How do they do? One thing they do actually is described here. They maintain their solute, right? The amount of solute in the cells is equal to the solute that outside the cells. So they try to actually balance out the solute inside and outside. If the outside of the cell have that kind of concentration of solute, they will try to maintain in the cells that amount of the concentration, which is comparable with the one that from outside. How do they do that? If you have high concentration, wouldn't it be affecting the any mechanism that happen in the cells? Therefore, they will have compatible, they call compatible solute. Because these compatible solids, including inorganic or organic, they wouldn't affect the bacterial, their metabolisms and growth. So their presence will actually balance out the concentration inside outside, but at the same time they are not affecting the vitamin, and other amino acids. Here we go. And so we talk about hairophile. The hairophile can be divided, of course, into different group classes. And how do they do? Depends on how much the bacteria can tolerate, or the antibacteria can tolerate the salt. So they get divided into slight hairophile, moderate hairophile, and extreme hairophile. Alright? Now we're talking about this river, which is the fresh water, have very, very little, because the river has very, very little, 0.05 of the salt. And those are the conditions that normally is non-hairophile. Those are non-hairophile will be able to live in this fresh water. But in oceans, how many percentage of salt? 3.5 normally. So if you actually has your aquarium at home, you're making your own seawater, you know that you have to put 3.5 grams of the sea salt into the water to make. Example, we sample the concentration of the salt in seawater, and 3 to 5%. So those are the slight hairophiles, and those moderate hairophiles, right, will be those the microorganisms that present in the oceans. Now, some of the moderate hairophiles can actually live in the cheese. Cheese has about more than 5%. Now, we talk about this extreme hairophiles. They can live in almost like saturated salt in the water, more than 35%, which is very, very high. How high is the salt in this saturation water? If you're making this, possible to make it if you heat up the water and dissolve the salt. But if you leave it at room temperature for 1 day, they form crystal because they are oversaturated. If you put into the fridge immediately, I think within an hour, they will form crystal. So it's highly saturated with salt. Now, this is a very common diagram that regarding the Y is the growth rate, how well they can grow in terms of the salt concentrations. Better here. So with the salt concentrations from in percentage, so from 5 to 20, those are not a non-hairophile, meaning they cannot tolerate any salt, such as E. coli. They only can tolerate 0.03 to 5%. Okay, those are the non-hairophile. And this is the hairotolerate tolerance, meaning the slight hairophile or hairotolerance. Staphylococcus aureus, they can tolerate until 11%, let's say. So they even can live the best into a very small salt or with zero. They're happily living there. So their growth rate come to the peak at low salt concentration, but they are still be able to have minimum growth at high concentration, as high as like 10% of salt. Those are Staph aureus. So Staph aureus, they are present on our skin. So why they happily that living on the surface of our skin? We sweat and we produce salt. So they can tolerate and they can live on our skin. And here you have hairophile, they grow, they can actually, the best growth is actually like 6-7%, but they can actually have a wider range and tolerate a higher salt concentration. And this is a group that most people focus and very interesting because at very more than 20%, they still maintain the growth. And this is an example is the Helicobacterium Selenum. Alright, these bacteria, they are rock-shaped archaebacterial. They're aerobic. They take oxygens. Their origins, I think, in 1970, around there. It was first discovered when they found them in the saltfish. Actually, it's not found in the nature, in the saltfish. And then later on, they found this Helicobacterium Selenum in Great Salt Lake in the United States, where you have a very high salt, and up to 25% of salt. Dead Sea, and also in Dut Lake, Anatolia in Turkey. So those are the lakes which contain very, very much of the high percentage of salt. And they also having a purple membrane because they contain some of the pigment in the membrane. So there is, you see that purple color, that entire lake, half of them at least in purple color. That means they are full of this antibacterial. Now, we want to look at that, how this Halobacterium selenatum, they can adapt to the high salt conditions. We look at them one by one. The one that I highlight, right, you need to pay attention, meaning that they're using the strategy 1, 2, 3, 4, 5, 6, 7, 8. 8 of them to adapt to the high salt environments. Okay, which one we should look at first? We're looking at this first. They actually they are aerobic, but they also can do photosynthesis. In order to do photosynthesis, they have this bacterial rhodopsin. Bacterial rhodopsins, why people start to have interest of this particular antibacterial? Because they found this very interesting. Rhodopsins also present, the homolog also present in human. Where you can find rhodopsin in human? Actually in the eye, the retinal. Our retinal has rhodopsin, which is a pigment in the pigment cells that absorb the light. Same thing, this particular homolog, rhodopsin, homologous rhodopsin, also absorb the light, even the energy, to this rhodopsin to pump the proton out of the cells. So when they're pumping out of the cells, what happens is that you have accumulate the proton concentration high, and through the gradient concentration of gradient, then you will actually, the flow of the proton will coming back through the ATP synthase. Because high concentration proton outside, low become inside due to the pumping out by this bacterial rhodopsin. So when they come in, they generate energy. So there they have these bacteria using photosynthesis to generate the energy. Eventually, this is important. Right? Now, how do they actually, because they are those living in the lake, let's say that in the lake, and normally they need the sunlight. How do they need to get onto the surface, at least close to the surface, to get the light. Then they have the flagella. Flagella will help them to move to the surface where you can actually detect the light. So they allow them to move. Secondly, they have this gas vesicle. Gas vesicle is this kind of thing. Inside this vesicle, they contain gases. So allow them to flow to the surface. They use this kind of mechanism to get the sunlight. Now, we also look at this. This one is the potassium syncoder, which is a protein channel to pump in the potassium. Why this bacteria need potassium? Because outside there is a lot of sodium. They need to actually get a balance, because too much of sodium, iron outside, then the water will start to flow out. So they want to get a similar concentration of the iron, so they use the potassium. They pump in the potassium, and potassium is high enough in the intracellular and is balanced out whatever the concentration of sodium present outside. So therefore, there's no water going out. So they have the potassium syncoder to pump in potassium. And the potassium, of course, is one of those called compatible solute, are not affecting the metabolism of the bacteria. They are present a lot. It's just for the purpose to get high concentrations and make sure no water flowing out. After that, we look at which one? Alright, here. This is the membrane. And the membrane You know the antibacterial has the lipid bilayer membrane, right? They have the ether, lipid-based bilayer. Unlike the bacteria have ester, link, phospholipid. They have the ether link phospholipid. This is more able to tolerate to the tough conditions, not easily that is being damaged. So they have this. Plus, antibacterial has an S layer. S layer is actually a form of the protein that cover the surface of the cells. It can prevent any dehydrations, right? Because under all this environment, if you have a high salt concentrations in the lake, meaning that you have a very strong sunlight, otherwise the water will not evaporate and make the salt, right? And because high, very strong sunlight, indicating that no cloud. When there's no cloud, high UV radiations in this area. Now, they will actually, because it's very hot, so it prevents dehydrations, okay, through the membrane productions. And here, you have this one particular protein called bacterial rubellin present in the membrane. What is the purpose of that? This particularly can protect the cell from the UV radiations. And they have the so-called like a pink color. So when you blow on the colony, you see the pink color. And they themselves, this bacterial rubellin, are not like protecting a shield, not like that. They as a target of UV light. So they absorb the UV light, and then they reduce the absorbance of UV light from any other component. Right? They themselves actually do as a absorbing more UV light, so that they reduce any UV light absorbance from any other part of the cells. So this is for UV protections. Which one? This one. This is the nucleoid. Here says 68% GC content, meaning that the DNA has more G and C guanosine and cytosine rather than adenine and thymine. Why? Why high GC content? The only purpose is to make it stable. GC, G is paired with C, right? With 3 hydrogen bonds. Rather than AT, AT is 2 hydrogen bonds. So GC have 3 hydrogen bonds is become more stable by having too many AT. So it's stabilized and not easy to get the chromosomal DNA denature. Alright, the last one. Do I still have the last one here? The last one. This is talking about the protein, the enzyme and protein produced inside the cells. They tend to have more those aspartic acid or glutamic acid, which is having a negative charge amino acid present on the surface of the protein. They have high percentage of these negatively charged amino acid, and also they present at the surface of the protein. And this is the reason is that when the protein present in the high salt environment, more or less the protein will start to aggregate. In order to make the protein not aggregate is to make the protein soluble. How to make the protein soluble? By having this negatively charged amino acid on the surface of protein, this negatively charged side chain can interact with the positive molecule in the cells, and there it make it soluble and stop it from aggregations. But when it's aggregate, it cannot work. So this is how this Heterobacterium celanarums, and they take all these possible strategy to survive in the high salt environments. Okay? I saw somebody sleeping just now. They're sleeping very well, I think. Okay. Do we have reaching 1 hour or not?
Yes.
Almost. Let me finish this one. I think this is the last slide. Yeah, this is the last slide about the aerofoil. So we talk about just now the water availability. availability.
What the fuck?
So the water availability is different from the amount of water present in the environment, whether because it's the availability for the bacteria to use them.
It doesn't bounce
And
either.
do not, so therefore do not confuse with the presence of water, because the salt or sugar that in the environment, they can absorb the wet water and reduce the water availability. So when you have a pure water, normally we say is 1. Alright? So there are therefore a different type of that aerophile. So non-aerophile will require the water availability 1, meaning that pure water present in the environment. There's no other solute. Marine, which is a slight aerophile like Pseudomonas, some 0.98, you can tolerate. This is the best. And moderate aerophile, they can actually reduce the water availability. They require 0.91. And for extreme aerophile, they can have tolerate with more solid present environment, but then the water availability is 0.75. So it's actually reducing the water availability, and they are still living happily in this environment. Alright, I think the next slide is different. The next slide is pH. Okay, you can have a break. How long you want to have a break? 1 hour. Okay, I'll see you after 1 hour.
So looking down you die. Yeah. Fishy. Mà leo dám mà. Bự chi, leo什么bự？I said it because it was funny. 我比较喜欢那个三个那个小小个缆车。Matilda, this is a old name, old person's name. 不知道是哪一只。简单。也没有嘛，不知道。First one, what? Hit me on the first one. It's your phone, darling.
Yeah, darling.
Oh, so dedicated with Instagram.
What's that? See. I forgot already.
I mean, like, it's just Japanese. It's like Japanese. Like, no, like, it's like Japanese. I think your boyfriend
I think they died, yeah. Patience is key.
I know you are. The distance is. I think it's too steep.
太靠近太阳会爆嘛，对不对？
Well, let's see. My physics don't really work. So I thought
You say already, thank God already.
it's a waiting game. I'm stopping.
是啊，是。哎
So must be sending my
600K， OK， 到我了。
Is to ask the people not to talk to No, you never try before, huh? You come and try? You go the silent zone there and everybody start talking.
Okay, no, in the silent, like outside library, like the book there, like okay, I think, but inside the silent zone, I think.
No, the whole library is not supposed to be talking.
I see any NFL card, I see any. Malaysian dash experience.
Oh, I just know these are like, hella funny.
Hella funny. I know what's happening here. Ain't no way you're fucking using this. Oh my god, you lost.
There's no way. What? It's such a scam. Oh, how do I restart? Fuck.
You know, there's like this meme,
Wait, what the fuck? Look at this shit.
like the two friends.
I thought okay lah.
And then like,
You cut all the way to the
I have
fucking
for-
right,
for
bro.
Tario
What the
for
fuck? And then kind of like, oh shit. Bro, what the fuck?
Oh, you never look. What the
I mean,
fuck?
okay, the thing is,
Yeah.
who expect the car
Yeah.
turn like that, you know?
Yeah.
It's like a turning lane.
The guy turning never look. He just turned.
Yeah lah.
You think he signal then
Rose is.
his mother wrote.
Be careful.
Remember you got drive like this or what?
Be careful when you drive again. Don't be like him.
Why you want to say that don't be like him?
You see what you do?
He's not a bad driver.
We hit it first, son.
But it's not even first. I mean, it's hard to do if the intention wasn't that what then the follow-up question is that his intention wasn't that. But then it's different thing. And then the follow-up question is changing everything.
Boom!
Are you winning at least?
Um,
Damn, new high
Easy lah,
score.
because it's easy.
You
Everyone
high
also know it's easy, right? For kids one.
yeah.
Every versus for kids.
Yeah.
There's no way you fail this easy.
Yeah.
There's no way.
Yeah.
There's
And
no
I
you
haven't
go.
been failing.
Oh, you haven't been failing.
I haven't been failing.
Break all the records.
Yeah. Bird key. Then I got a bird. Okay, this one.
Why not do the fat one?
This one spins.
Oh, that, oh, yeah,
But the big one is a bad one. The big one do. It like
He just stays there. He's like a
No.
bitch. Oh no, he becomes big.
he expands. 他 expand 的。很 cute 欸。
This is where they make it.
But like it's really weak though.
But yeah, but really.
Yeah, but it's against for like ice.
Oh, things eyes.
Yeah.
Okay. Got the game all down.
Yeah. It's for kids, and I played it as a kid.
Yeah.
You know,
Like yesterday.
You know, my dad was addicted to Angry Birds 1.
Easily.
Chronically,
现在还有少女神一个 to Angry Birds 2。
In the jeans.
inherited.
去年 jeans。我屁股痒啊，我去帮你。
那就打过不一样。Blacko hit it first. Let's go.
I almost done already.
太迟了，
How many more? 8 more steps.
你原来 critical。OK 到了。
Why you backseat gaming? You got 3 stars. New record.
I can say every round. Holy shit.
Yeah, because this is the first record.
So the pH is also important because the environment can be acidic or
Did she forget she has a mic?
right, especially when we talk about food. So the food, bacteria like to grow in the food, which has a lot of nutrients they can take. Therefore, pH is rather important for certain type of bacteria. And it all is about the hydrogen ion concentrations. You have very high hydrogen, you have
Subway
alcohol,
Surfers.
not that you have alcohol. So they have a certain pH range that consider where they are the alcohol or alkaline form. And those who are the first one, those who are living at pH 7, the best, which is they have the maximum growth. They are at pH 7, which is neutral. So those neutrophils are suitable that grow at the pH of 7. So of course, if you have slightly acidic or alkaline, it's okay. But it's just the growth is dramatically drop a bit. And alcohol, as you can see, those of microorganisms has really a range from even as low as pH 1, right, and then up to pH 5. So anything that below pH 5.55 we call ester form. And those for alkaline form that they living at the pH from 8 to 10.5, which is roughly here. So 8 roughly around this distance. So you have to go about that. So here is another table showing some examples. For example, those very common bacteria, the E. coli, Staphylococcus, and Salmonella, those we commonly see, and they are neutrophil. And of course, if you have the sulfur oxidizing, such as Staphylococcus and Ferroplasma, these are the archaebacteria. They are living in the acidic conditions. And the alkaline conditions which contain the Vibrio, Torreria, and also some of the, this one, Necrobacterium in some of the alkaline lake and high pH. Now, there's another diagram showing example. Staphylococcus and Ferroplasma, both are archaebacterial. How do they adapt to the low pH? So when you have low pH, meaning that you have the hydrogen ion a lot accumulated outside the membrane. So your membrane have to do something. First of all, you have to use the energy to pump the hydrogen ion, the proton, out of the cells. Right? So because they keep accumulating the hydrogen ion, that's why the first thing is that there is some pump that allows them to pump the hydrogen out so that they can keep their pH in the cells, which is the cytoplasm, which is higher, not too low. Secondly, come in those potassium pumping into the cells. So when you have more potassium into the cells, and we know that potassium not affecting the physiology of the bacterial cells, therefore when you have more of this potassium, actually this potassium can displace hydrogen ion in the cells. So it's another way, indirect way, rather than directly pumping them out, they have something that will be occupied in there, the ion be occupied inside the intracellular, and then to displace them more, not allow more of the hydrogen ion to come in. Now, they also have something which is here. This is they have the cytoplasm, they have some so-called buffering molecule. As soon as you have the hydrogen ion coming into the cytoplasm, they will be taken and converted into something which is neutral. So they will not have accumulate a lot of hydrogen ion by itself in the cytoplasm. And this one as well, this is actually the lipid bilayer for the archaebacterials. If you remember, we talked about the archaebacterials. They actually can form, instead of double layer, the bilayer, they have monolayer like this. So it's actually, these look like the top layer and the bottom layer, they fuse together to form a monolayer. These monolayers, it has a purpose to make the membrane become impermeable for the hydrogen. So the hydrogen is not easily coming in, right? And you also, because of the very high, very low pH inside the cells, is easily under the acidic condition, easily is damaging their DNA. So they will have a very good DNA repair system, actually to repair any damages on their DNA. And here, similar to the aerophile, they also have the protein present in this kind of astophile. They also have the acidic residue that present on the surface, so that they can actually interacting with their hydrogens that make it more stable. Because hydrogen is positively charged, if they have the surface has more positive charged amino acid, they likely can cause the protein to aggregate. So that if they have placing this acidic amino acid which is negatively charged on the surface, they can actually making the ionic interactions with their hydrogens, which is stabilize the structure of the protein. Alright, I think more or less that's all that has been mentioned. Oh, this one is a Helicobacter pylori. Helicobacter pylori is a neutral form, meaning that it doesn't like low pH, it doesn't like high pH. It likes pH 7, neutral. But you know that Helicobacter are living in So this is how this bacterium they can survive by actually generating an immediate environment with their pH 7. Okay. Now temperature. Temperature is rather simple. Temperature, they cannot control the temperature. They cannot regulate the temperature in the intracellular cytoplasm. So that temperature from the environment can have immediate effect on the enzyme functions because all the protein, as you know, normal protein in general, protein, if you raise the temperature to 65 degrees Celsius while you are cooking, you're damaging the protein. So protein cannot tolerate high temperature. But somehow, this bacteria can actually maintain the protein structure. So membrane integrity, the membrane actually can be damaged, right, at high temperature as well. Even the DNA, when you have high temperature, they start to denature. Double-stranded becomes single-stranded. That is how the temperature can damage the cells. I think there's talk about. Please look at this particular diagram. This diagram indicates that the growth rate, so the temperature for most of the here depends. Let's say that those very common, most bacteria is 7 degrees Celsius. Right, if they are the optimal temperature at the right temperature to be 7 degrees Celsius, the best for the most bacteria. But of course, when you increase the temperature, they can survive and they still can grow, but in the minimum growth rate. And if you actually reduce the temperature, so this is the minimum temperature they can survive and they still can grow in the minimum growth rate. That's about it. Okay. So what do we call those to the moderate temperature? So which one is moderate temperature? Let me see. Mesophile will about the temperature, room temperature to 45 degrees Celsius. So this is the most general, most bacteria living in this temperature. And those are the normal human microbiota or human pathogens, E. coli, Salmonella, Lactobacillus, they all are mesophile. Cyclophile, on the other hand, those can tolerate low temperature. We call cyclophile. So they are flow level, and what is they're ranging the temperature they can live -5 degrees Celsius to 20 degrees Celsius. We also come in here, except cyclophile, mesophile. And mesophile is actually in this 37 degrees Celsius, the best. And if you have cold level, you have cyclophile, which is from -5 to 20. And you have between mesophile and cyclophile, you have these cyclotrophs. Cyclotrophs can tolerate slightly low temperature. Their range is from 30 to about 0 degrees Celsius. So, what does that mean? You have encountered all these cyclotrophs. Those bacteria that can grow happily, okay, and in your fridge, right? And they are the cyclotroph. Those bacteria living in the fridge are cyclotroph. Now, if you have temperature, high temperature, we also have bacteria like high temperature. We have thermophile, hyperthermophile. So this is the extreme, more than 100 degrees Celsius. This can tolerate up to 80 degrees Celsius. And remember, these have a different name. Can you remember all these names? At least to get, you may not need to know the spelling. Probably better to know the spelling. The reason I'm saying that
Probably
it
better
comes
to
from
know the
your
spelling.
MCQ. So it doesn't require you to write and spell out the name, but you need to know what are they. Those questions that come from MCQ. Alright. Now we talk about the adaptation for cyclophile. Those they like cold.
Boring.
the fluidity of the membrane. How do they do that? They have unsaturated fatty acid. What does that mean? If you have a fatty acid which is strict, it's called saturated. If you have the fatty acid side chain, right, which you have a pink, this is
Evidently.
unsaturated. So you're comparing your fatty acid, you have a pink, this is unsaturated hydrogen bond because there is a double bond. I think here's showing a double bond because of the formation of double bond. So the change of the directions of the structure, and you have a kick here. So the king, as looking, that some of them have this. So they actually having it like separating them in a certain distance from each other. Rather than if you do not have this king, they actually align them very organized in this structure. So imagine that if you under the cold temperature, this particular membrane easily is frozen, is actually can easily damage. Rather than you make it more fluid, having the kick, right? So you make it more fluid and able to maintain its structure at low temperature. So this is first thing. Second, they have those cold shock protein. They call cold shock proteins. Those protein, I didn't have a diagram here, but those proteins basically they are used to protect the nucleic acid. So for example, those cold shock protein, when suddenly you reduce the temperature, even from mesophile, they have those cold shock protein. What happened that, let's say the mesophile, if you suddenly put them in the fridge or freezer, they immediately will express the cold shock protein. And this cold shock protein will bind to the nucleic acid and protect them and not allow them to form a secondary structure. Let's say here, example is the RNA. RNA in the bacterial cell need to be transcribed, right? So if they form a secondary structure, especially there's a tendency is under low temperature, they form a secondary structure, which is ideally will prohibit them to go for transcription. So when you do no transcription, no translation, no protein is made. So the entire metabolic system collapses. And those cold shock protein just to protect the structure of the RNA here. This is more interesting. Antifreeze protein. And what is antifreeze protein? And here is showing that this particular protein, if you imagine your cytoplasm, right? The cytoplasm full of solids and water. So if you put the cells in the freezer, it will be frozen, including everything inside the plasm. Now, in the cytoplasm, in order not allow it to be frozen, you have this protein called anti-freezing protein. This protein not bind to any other protein, but bind to the ice crystal. And it binds to the ice piece, and then they put ice in it, but it's the ice. They binding on it to prevent the ice forming a bigger crystal. So this is a way to prevent the frozen, the freezing in the cytoplasm. Because the freezing is start with forming this ice crystal, a little by little, and then they all become all this ice crystal. Right. The next one, I think I have the next one called the thermophore. Thermophore is simple. Thermophore, those are living in high temperature, even up to 80 degrees Celsius. And the one that in the Yellowstone, the thermophore, they actually isolated this called Thermus aquaticus. This is bacteria, Thermus aquaticus. Familiar with this? Because when the person actually get a Nobel Prize because of the invention of polymerase chain reaction, which is a technique that we are still using it now. Actually, all is rely on the discovery of this Thermus aquaticus from the hot springs. Why? Because they survive, they die. They are necessary for them to live in the high temperature. And I think there are more explanations coming. And this is a hydrothermal vein that actually they also have a lot of bacteria. Either they are living in a symbiotic with relationship with others organism, or they are living in. I think they don't living by themselves. Not many are living by themselves. They are symbiont that with other organism, and you know that those are the gases coming from underground, which is extremely hot, can go up to 300 degrees Celsius. So around the hydrothermal vein, you have around at least more than 100 degrees Celsius. Alright, so those bacteria normally they are, because they are actually under the bottom of the sea, so they do not have any sunlight, so they are chemosynthetic bacteria. A lot of, because when this coming out, right, this is all the gases coming out from that, they bring a lot of metal. So the metal will precipitate around this thermal vein. So they can become a chemosynthetic bacteria, utilize it as a source of energy. Did I miss anything? No, this is the next one. Alright. So all of this, only one thing they have to do. The protein, the DNA, RNA, and the membrane, the three main components, they have to be stable and function at the high temperature. So how do they do that? First of all, of course the membrane has to be strong, which is can actually endure any high temperature. So you have this ethyl ring and acetylene. Acetylene in the most common bacterial and eukaryote, but in those antibacterial, they have the ethylene. Right? This is one, and this is a vitamin side chain. Vitamin side chain is something that actually extended from the fatty acid. So all these eukaryotes, the bacteria or bacteria, common bacteria, they do not have the vitamin side chain. This vitamin side chain standing aside to make everything is some interactions with the fatty acid tube to each other, so that they are alive and strongly not vulnerable to the temperature. And furthermore, if just I mentioned some antibacterial, they have this tetraether monolayer, so that they actually stabilize the membrane by having only a layer rather than you have two layers. So this is how they stabilize the membrane. Any others? Yes. Now, they're also using for the DNA. One example is the reverse gynase. Reverse gynase is topomerase. You heard about topomerase? DNA is supercoiled. When you want to open up the DNA during replication, you need to uncoil the DNA, right? Open up the DNA. So that is topomerase. But this is a reverse gyrase. Reverse gyrase is doing the opposite. So they make positive supercoiling. During DNA replication, open up DNA is negative supercoiling. But this one is a reverse. So what to do? In high temperature, DNA tend to denature. What happens is that if you have reverse gyrase, actually it can actually make it supercoiled again. So putting it back into the positions. Right, not allow it to denature all the time. And of course you have all these heat stable enzymes and proteins. So you have those amino acids which is more stable, such as aspartic acid, asparagine crystal, and glutamine. So in the protein composition, those are the amino acid which is not more stable. And you also has increased the hydrogen bonding between the amino acid in their protein. So they need more interaction to make the protein compact. And of course, most protein, they have the hydrophobic core inside. You have the protein. At the center of the protein normally is hydrophobic. This hydrophobic in the and slow the unfolding process. Alright, to stabilize the protein, you have all this structure that in your protein. So the protein from thermophile, when you compare with the protein from mesophile, you've been wondering, is the same protein, they have the same amino acid to make up the protein. Why that one is denatured easily when you increase the temperature, but in the thermophile, those proteins are not so easy. I think they have this bit and that, and a bit of this property make them tolerate to high temperature. And in general, you will see the thermophile protein, they are much more compact, meaning that they do not have any space. Because you have folded the protein, right? You will have space in the protein. But those thermophile protein, they are highly likely you couldn't find more space, not many space, so that they are more compact and stable.
I don't think it's gonna happen.
Okay. Come to the oxygens. This is also interesting. I think you're familiar with this. That glycolic tube. Now, oxygens. Some bacteria require oxygen, some bacteria not require oxygen. So oxygen is one of the important components for the bacteria. Then we have a terminology for all different type of the oxygen lover or non-lover. Now we start with the opicate aerobes. So when you actually prepare the medium, adding the diglycolate is one of the reagents that adding into the medium, and actually it will remove the oxygen out of the medium by adding this diglycolate, right? So in the tube without the oxygen, now then we will actually create the growth of the bacteria at different level in the tube, and then depends on where do they grow, right? Then we can actually define them whether they are opicate aerobes or opicate and aerobes, facultative and aerobe and whatsoever. So we look at the first one, opicate aerobes, meaning they are strictly using the oxygen. So they cannot grow without oxygen. They utilize oxygen. Opicate and aerobes, they totally cannot tolerate oxygen. They can put out oxygen. Facultative and aerobes, what does that mean? They use oxygens, but they can grow without oxygens. They can survive without oxygens. So this is the opicate aerobes. They are actually, you found that the bacterial growth is denser at the top. Why? Because here there is the oxygen diffuse. Although you have been removed the oxygens by this diglycolate, but eventually oxygen, if you are still not, you are not like closing the tube, you still oxygen diffuse. So therefore you found the bacteria hunting for the oxygen, and they are growing at these regions. Opicate and aerobe, they totally cannot tolerate oxygen, so we found that at the bottom of the tube. Facultative and aerobe, they utilize oxygen, but they are not really need them to survive. So they actually have more of them here, but slightly less dense at the bottom. Now this one, tolerance and aerobes. What does that mean? These are the ones that they do not need oxygen and also not killed by the oxygen. So whether they do not need oxygen, meaning that they can grow anywhere, and they are okay, they are also fine in the presence of oxygen. So they can everywhere, you found them growing in the tube. And you see the difference of this and this? Yes? One is using oxygen, the other one is not using oxygen, but both are comfortable with the present or absence of oxygen. The last one, microaerophile. oxidase here, you don't see a lot of cells. So you have to reduce the oxygen which is lower at the lower bottom here. You can find more bacteria growing at this region. Okay, those are the names that you need to remember. This is another slide telling the example of the what are the obfuscate aerobes and which one is the obfuscate and aerobes, fertility and aerobes, and aerotolerance and multiple aerosol. I think you can take a look of them. Right? This is an example of the bacteria. Now, come to this. It will explain later this. Let me explain this first. Then I will tell you why this is important. For the bacteria, when we define whether it's strictly aerobes and this is aerotolerance or not. Now, you heard about reactive oxygen species, ROS, right? Everybody talk about ROS because ROS is very toxic to human. If we accumulate a lot of ROS in our body, basically it will damage our organ or tissue or cells. So, same things. The bacteria also, when they utilize the oxygen, they also generate this kind of reactive oxygen species. So, one common property of the ROS is that they are very reactive. They are very reactive. They can actually steal the electron whenever they around them. So when you actually have these radicals, a reactive radical around the tissue, they actually steal the electron out of their cells from the tissue, and in that way they damage the tissues. So in here, your oxygens, when they take an electron, it will generate this so-called superoxide radical. So this superoxide radical, when you're actually using the enzyme called superoxide dismutase, you can convert the superoxide radical, which is this one, into hydrogen peroxide. Hydrogen peroxide, which is less toxic than the superoxide radical. Alright? Now, then you can use this catalase or glutathione peroxidase. You can change it into water, or you can undergo another pathway called Fenton pathway, or you can Haber-Weinz pathway, and you can generate hydroxyl radicals. So all these are ROS. And the bacteria, they actually producing this kind of enzyme as well. You will see. This one. So this is just explain each of the enzyme. Superoxidase dismutase. Then they actually convert the superoxide radical to hydrogen peroxide. Catalase is an enzyme, bacterial enzyme, can actually convert the hydrogen peroxide into water and oxygen. So peroxidase is another enzyme that in the bacterium can convert hydrogen peroxide into water. So if you look at that, this is explanation, this is summary of the pathway. And how it goes, if you have this ROS, right, it will turn that this superoxide radical through the superoxide dismutase, and you generate oxygens and hydrogen peroxide. Be careful, there's oxygen here. And with that, if the bacteria have peroxidase, the bacteria can further actually convert hydrogen peroxide into water. But if the bacteria have catalase, it can convert the hydrogen peroxide into water and oxygen. Now, those enzymes can be present in the bacteria, but not all bacteria have these three enzymes. If you look at these obfuscate aerobes and most of the facultative aerobes, because they require oxygen, so they will have this superoxide dismutase, first of all, to convert this superoxide radical into hydrogen peroxide with the oxygens, right? So they have superoxide dismutase for these obfuscate aerobes and facultative aerobes.
they will not have go to this pathway. Instead, they will use this catalase which they have to convert more to water, which is not toxic and also generate oxygen. So therefore, they have superoxide dismutase and catalase. They do not have peroxidase. Now, for those aero tolerance and aerobes, do you remember what are these aero tolerance and aerobes? Let's go back. This aero tolerance and aerobe, right? Meaning that they do not need oxygen. They can, but they are not. Oxygen will not cause any toxicity to them. So those are this category. What do they have? What they have is that they have this superoxide dismutase. So therefore, that's why they are not, the oxygen are not toxic to them, because they can convert, right, all this superoxide radical into hydrogen peroxide. And further, they have this superoxidase, then they can convert it into water. Because they do not require oxygen, they are not the aerobes. They are not aerobes, so they do not require oxygen. They do not have catalase, but they have this peroxidase, which actually to convert all these raws into water. And they do not afraid, because of the presence of oxygen can generate all the this radical the ROS, sorry. They can actually generate toxic ROS. So they are having the enzyme to breaking it down into something that is not toxic. That's why they are aerotolerant and aerobes. If obligate and aerobes, meaning that they cannot live in the presence of oxygen. The oxygen will kill them. And the reason is why they do not have all these three enzymes. So no way they can convert these radical species into something that are non-toxic. And because they do not have this enzyme. So therefore, it really depends on the presence of absence of enzymes in the cells. They actually can divide them into different classes. Good. Okay. Lastly, this one is only have one slide. Just to tell you that some of the, not very important, but some of the bacterial, cyanobacterial, as we always mention, has used light to generate the energy. So they have actually, the energy from the sunlight actually can be absorbed by the pigment. Carotenoid is one of the pigment, color pigment, that also present in some of the bacteria, that they can convert into the energy and later glucose. Basically, we can divide them into two categories. One is photoautotroph, right, and the other one is photoheterotroph. The example of photoautotroph is the cyanobacteria or green algae or the green sulfur bacteria. What they do is that they actually take the sunlight, right, and then they use to generate carbon dioxide, and with that they have converted into glucose. And for the photoheterotroph, they are not using carbon dioxide, but they do another pathway, but they require the sunlight. They actually degrade the organic compound to making as a nutrient for generating the energy. Those are the photoheterotroph. I think such as sulfur bacteria, they are the proteobacteria, and they also need sunlight, but they are using the organic compound as a source of nutrients. Okay. That's all for today. Yeah. Good.
什么家？
So you may
Of course,
any questions? Then we have